Okay, so welcome back to this next video in which we're discussing light microscopy. Okay, right. So we're in the process of discussing lenses. Now, I was just about to explain to you why when you look for a magnifying glass, you don't generally see the image upside down. And the reason is, firstly, that the distance between the thing you're trying to look at and the lens is too small, and also the distance between the lens and your eye is too small. So if you imagine putting your eye here, so let's put your eye here, just put a circle to represent your eye, okay, rather than, you know, where this image is being formed over here, what basically you'll see is you won't see it upside down, basically, because the light hasn't yet had time to cross from one side to the other. So basically, you will still see this side at the top and this side at the bottom, because the light that will actually be coming into your eye from this top portion up here will be, you know, the light from like here. Okay, and the light coming in from the bottom will be the light from here, basically. So basically, it's because you're not positioning yourself and the thing you're trying to focus in the correct lengths away from the lens in order for it to actually work, basically. And you can play around with a magnifying glass. You can, you know, if you make the distance between the magnifying glass and the thing you're trying to look at large enough, and you make the distance between the lens and your eye large enough, you will be able to turn the image upside down, and then this will be occurring. Right, okay, so basically we're going to use this principle in microscopes. Okay, so what we're going to talk about is compound light microscopes. Okay, and compound light microscopes contain two lenses. So they contain what's called an objective lens, and then they also contain an eyepiece lens. So you have one lens here which is called the objective lens, okay, or simply the objective. And then you have another lens further back, which I'll put here, which is then called the eyepiece lens. So this is known as the eyepiece lens. Okay, and these lenses that you are going to use in microscopes will be tiny. They will be absolutely tiny compared to the lenses that we have in magnifying glasses. So basically, you won't need massive distances in order for the turning of the image upside down to actually occur, basically. You'll need tiny little distances. So this turning of the image upside down is going to occur, basically. Now, what's going to happen, the principle of what's going to happen, is that the slide that you're trying to look at will be here, okay? And the human eye will be pressed against this eyepiece lens, okay? So let me draw the human eye sitting here. So we'll have a look at the structure of the human eye. Okay, right. So, let me put the human eye here. Okay, so this is the sclera, this outer layer, and the sclera is continuous with the cornea at the front here. Right, so let me draw the cornea and the sclera first. So this is the cornea at the front here, and then we have the sclera coming back here, okay? Like so. And it goes all the way around. Okay, now, at the front here, what we have is two large muscles, which I'll put here, one like so, and then another one over here. Okay, and these are called the ciliary muscles. Okay, so I'll colour them in a red colour to make it clear that they're a piece of muscle. Okay, right. So these are the ciliary muscles here. Right, so there's one, and there's the other. Okay, and then what you're also going to have is the iris. So I'll put the iris here, okay, which is also a muscle, okay, and this will have a hole in the middle at the center, which is the pupil. Okay, so these two extensions of muscle further inwards, these are the iris. Well, this is the iris. Okay, right. Now, after the iris, behind the iris, what you then have here's the lens of the eye, and this is a third lens that you need to consider when we're talking about the microscopy. Okay, and the lens will have suspensory ligaments here, like so, that are holding it to the ciliary muscles here. Right, okay, now we need to also put on the retina, so I'll just complete this drawing now with the retina here, going all the way around. So this is the retina. Let's label up our drawing. So this is the retina of the eye here. This outer layer is called the sclera. Okay, we've already done the ciliary muscle, the iris. Let's have the lens here. Okay, then we've got the suspensory ligaments here. Okay, suspensory ligaments. And then we've got the cornea at the front. So remember, the sclera is the name for this portion when it's at the back of the eye, but at the front here, it's called the cornea rather than the sclera. Okay, so I'll label that one up next. And actually, I might not, well, I'll try and squeeze it in. I'm just running out of space is what I'm a little scared of. Okay, so this is the cornea. And then you have 3 separate chambers. Okay, so you have this chamber that's in front of the iris but behind the cornea, which is called the anterior chamber of the eye. Then the chamber which is behind the iris but in front of the suspensory ligaments and the lens. There is a space between the iris and the suspensory ligaments and the lens. That's called the posterior chamber. And then the chamber behind the iris, behind the lens, behind the suspensory ligaments and behind the ciliary body, all of this in here, this is the vitreous chamber and contains the vitreous humour, whereas the anterior chamber and the posterior chamber, which are continuous with one another through the pupil here, they contain aqueous humour. Okay, right, so the human eye lens is designed to focus light that is coming from infinity, basically. So let me just explain what that means to you. So basically, if you have light that is coming horizontally, i.e. that is not coming from a point source, so if you have light like so, okay, that is coming horizontally, what can happen is that this can be focused by a lens. So let me draw the lens here. Okay, and you might notice that there's some incredible symmetry here, that you can actually guess how this is going to be focused. Okay, because if you've got light coming like this, how is the lens going to focus it? Well, this is the exact reverse problem to a problem that we've already considered. We've already considered the focal point, basically, which, remember, was that point that if you had that point sending out light, all of that light would go through the lens and then be refracted so that it was coming horizontally. Well, basically, now we've got light coming in horizontally, and what's going to happen to it is that it's all going to be focused down to the focal point, basically. So all of the light that's coming in horizontally will be focused to a single point, and that point will be on the back of the retina, basically. Okay, so here is a focal point. So you might say, well then, is all of the light that's coming into the eye being focused onto one point of the retina? That's insane. No, is the answer. Light that comes in at a different angle, because not all of the light is going to come in horizontally. Some will come in maybe at an angle like so. Okay, so if we have light coming in at this angle, okay, this will be focused to a different point on the retina, basically. Okay, so this will be focused to some different point on the retina. So let's draw this happening. So it might all be focused onto this other point of the retina. And I realise I haven't really shown it like it would be in the eye, because the retina is far too close to the lens. But basically what's going to happen is, if we imagine the retina here, when the light comes in at different angles, you're going to get it being focused onto different points of the retina. But the human eye lens is designed to focus this light that is all coming in horizontally, basically. So it will focus light that's all coming from a certain point, you know, far, far away. Okay, so we need to take that into account when we build the microscope. So back to our drawing of the compound microscope. So basically, let's start with the objective lens. What the objective lens is going to do is it's going to hugely magnify the image. Basically, it's going to do exactly what we have here, except that we are now dealing with some microscopic slide rather than a highlighter pen. Okay, so let's say we again have 2 points though. Let's say point A is here and point B is here. Okay, and then there's some portion in the middle here. Then what's going to again happen is light coming from point A will be focused by this lens. All the light coming from point A will be focused, and the point to which it's all focused will be some point maybe down here. Okay, so here's the image of point A. Okay, and then it will be the same for point B and all of the points in between as well, of course. Okay, so here comes all the light from point B, and it might, all be focused up to some point over here, which we'll call the image of B. Okay, and that will have hugely magnified the image, basically. So now you'll have a bigger image, just like the highlighter pen, basically. The image here will have now been turned into a bigger image that is ready to be seen here, if only we could actually see it. Okay, now what's going to happen is that the light is now going to come through this microscope here, basically. Okay, sorry, through this lens here. So let me just continue this dotted line here. Now, basically, this is the eyepiece lens, and what you can do is you can change the distance of this eyepiece lens, and what you are going to do, you can change the position of this eyepiece lens on the on the microscope. Okay, so you can move it forwards and backwards. Now, why is that so important? Well, because we are forming this image here, this upside down image is being brought into focus on this plane here. Okay, now, basically, you do not want this lens to actually be on the same plane as this image that we're forming here, but what you want is it to be a distance from this plane which is equal to its focal length. Okay, so you want this distance here to be equal to the focal length for this eyepiece lens. So let's call this the focal length for the eyepiece lens, so I'll put an E there, and this might not, in fact it won't be the same as the focal length for the objective lens. But the eyepiece lens, you know, you'll take into account its curvature, its size, and you could work out its focal length, basically. And what you're now going to do is modify the position of the eyepiece lens so that the distance that it is away from this image that is being formed in this plane is equal to its focal length, basically. Now, why is that such a clever thing to do? Well, what do we know about lenses when you shine light from a point source into them, and that point source is a distance of the focal length? Well, basically, all of the light beams will be focused in parallel lines. Now, previously, we have only looked at the point source of light being actually at the center of the lens and sending out all of its light beams. And then what happens is the lens will, diffract them all. Sorry, not diffract, refract them all, so that they're all running in parallel. Now, what happens if the point, is somewhere up here, but is still the focal length from the lens? Well, let me draw that out over the way, because obviously we're going to need that. Okay, so let's have a lens here. Okay, and let's say that that's the center of the lens. And let's say that this plane here is the focal length of this lens away from the center of the lens. And we're now going to take some point here, and we're going to have this as our point source of the light, basically. Okay, so what is going to happen? How is this, what is this lens going to do to the light? Well, basically, all of the light beams are still going to come out parallel, but obviously they're not going to all come out parallel and horizontal. Instead, they're going to come out at an angle. So let me show this happening. Okay, so imagine all these light beams rocketing towards our lens here. Okay, and now they're all going to come out parallel, like so. But they're no longer horizontal, they're going off in this direction here. Okay, and hopefully you might now start to see how this compound microscope is going to all click together. So we have moved this lens to the position where this plane, this imaginary plane that we've got here, where the image is being formed, well, brought into focus, the magnified image is being brought into focus there. We have now moved the eyepiece lens so that it is its focal length away from that plane. And of course, what's now going to happen is that exactly the process that I have just described is going to happen for these points. So this point, and now I'm only going to draw two of the beams of light. These will both be refracted off, and they will be parallel to one another, and they'll be going off in this direction here. Okay, and the image of point A, again, this will be sending off light. Okay, like so, and these will both be going off parallel, like so. And of course, I've only shown two beams of light, but of course there will be loads of them, and they will all be coming off parallel.